DBSCAN clustering. Density-based spatial clustering of applications with noise means DBSCAN is an unsupervised clustering machine learning algorithm. Hello everyone, welcome back. In this video, we are going to be starting the new topic of AI Developer Level 3, which is DBSCAN clustering. The main idea behind DBSCAN is to find out whether a point belonging to a cluster is close to many points from that cluster or not. It separates clusters of high density, means having many data points, from clusters of low density, having a few data points. We're going to talk about the required parameters. Ellipson, epsilon, which is E. So it is the distance that DBSCAN uses to determine if two points are similar and belong together or not. Minimum point means minimum PDS. The minimum number of data points within the radius of the neighborhood for the neighborhood to be considered a cluster. A low minimum points help the algorithm build more clusters with more noise or outliers. More required parameters. Now talking about the core points. Core data points have at least minimum point number of data within their epsilon distance. And the border points are on the outskirts as they are in the neighborhood but have less than the required minimum points. Now we have outlier points. These points are not part of a neighborhood and are not border points. The steps. Step 1 is to set the epsilon and the minimum point values. Step 2 to select a random point and check if it has the minimum points data points with the epsilon distance. If yes, then a cluster is made, else a point is considered as a noise. Now the neighborhood of the cluster is also checked. If they also meet the minimum points criteria with the epsilon distance, then the cluster is expanded. Else it is not. Now step 2 and step 3 are repeated until all the data points present in the DBSCAN are checked. Applications. Number 1 is scientific literature. To identify the research articles belonging to the same topic or discovery. Images of satellite to discover the abnormal patterns happening in the demographically via satellite. Crystal, crystallography of X-ray to identify the sets of conditions appreciated to make a crystal of a protein. Anomaly detection to find an anomaly or defect in the cells, temperature of a reading, flight scheduling, and etc. Benefits and the drawbacks. Advantages of DBSCAN. It does not require us to predetermine the number of clusters like K-means algorithm. It handles outliers like a champ. It can separate high density into small clusters. It can cluster nonlinear relationships, means find arbitrary shapes. Now the disadvantages of DBSCAN. It struggles to identify the clusters within the data of varying density. It can suffer with high dimensional data. And it's very sensitive to the epsilon and minimum points parameters. We're now going to move on to the coding part, which is going to be in the next video. So stay tuned till then.